Hi everyone, this is part 2 of the treatment of genetic disease. Please note that my lectures will have an AI-generated voice and an AI avatar. My computer can no longer record my voice, so I have had to use alternative method. I apologize. We will start this chapter by discussing enzyme replacement therapy. Enzyme replacement therapy has revolutionized the treatment of lysosomal storage disorders. For instance, V-glucocerebrosidase effectively normalizes biochemical markers and alleviates symptoms in Gaucher disease, though it remains costly and has limitations in treating neuronopathic forms. Adenosine deaminase deficiency, or ADA deficiency, is a life-threatening autosomal recessive condition affecting purine metabolism. Toxic purines accumulate, severely compromising immune function by damaging T and B lymphocytes. Without treatment, affected children typically succumb to infections within their first 2 years. While gene therapy and bone marrow transplantation are emerging as long-term solutions, PEGADA serves as an effective short-term therapy. PEGADA, a modified form of adenosine deaminase, improves therapy by reducing neutralizing antibody formation and functioning effectively in extracellular fluid. With an extended plasma half-life of 3 to 6 days, PEGADA restores enough immune protection to improve patient outcomes significantly. This therapy demonstrates how chemical modifications can enhance the clinical utility of enzymes, even when used outside their usual cellular location. Enzyme replacement therapy has transformed the treatment of Gaucher disease, specifically the non-neuronal form. Deficiency in V-glucocerebrosidase leads to the accumulation of glucocerebroside, causing anemia, splenomegaly, and bone damage. ERT effectively corrects these abnormalities, particularly when started early. Targeting macrophages through mannose receptors ensures precision, but this approach cannot treat neuronopathic forms of the disease. Despite its limitations and high cost, ERT has been a monumental success in managing lysosomal storage disorders. Now we will discuss modulation of gene expression. Modulating gene expression as a treatment for genetic disorders was once a distant possibility. However, advancements in our understanding of gene regulation have turned this idea into reality. As research progresses, this strategy is becoming a cornerstone of innovative therapies. Spinal muscular atrophy, a severe autosomal recessive disorder, results from mutations in the SMN1 gene, causing motor neuron degeneration. Nusinersen, an antisense oligonucleotide, enhances SMN2 splicing by promoting exon 7 inclusion, enabling the production of functional SMN protein. Since it doesn't cross the blood-brain barrier, it requires spinal injections for delivery. Clinical trials for nusinersen have demonstrated remarkable outcomes, with significant survival and motor function improvements across SMA types. Nearly all SMA type 2 patients could sit unsupported after 3 years of treatment, and over 75% of SMA type 3 patients achieved independent walking, underscoring the life-changing potential of this therapy. Risdiplam, an FDA and EMA-approved therapy, is a small molecule splice modulator targeting the SMN2 pre-mRNA. By binding to specific sites, it stabilizes the ribonucleoprotein complex and promotes exon 7 inclusion, increasing full-length SMN protein production. Its oral administration makes it a highly accessible option for spinal muscular atrophy patients. Increasing gene expression from wild-type or mutant loci is an emerging therapeutic approach. Histone deacetylase inhibitors, already approved for certain cancers, hold promise for treating monogenic disorders, particularly those with epigenetic origins. While still in early phases, this strategy opens new avenues for targeted therapies. RNA interference offers a powerful way to reduce toxic mutant gene expression. Small interfering RNAs target specific RNA sequences for degradation. This technology has shown great success in treating transthyretin amyloidosis, providing a first-line therapy for TTR polyneuropathy and showcasing the broader potential of RNAi for inherited diseases. Exon skipping is a therapeutic strategy where specific exons are excluded from pre-mRNA, potentially restoring gene function in cases of frameshift-causing mutations. Using antisense oligonucleotides, this technique has shown promise in treating Duchenne muscular dystrophy. For instance, skipping exon 51 can restore the reading frame in approximately 13% of DMD patients, increasing dystrophin-positive fibers and stabilizing walking ability in early trials. Genome editing, particularly with CRISPR-Cas9, allows precise changes in the DNA sequence. This technique creates double-strand breaks at target sites, which are repaired via homology-directed repair using a wild-type DNA template. A remarkable example is in sickle cell disease, where editing the BCL11A enhancer sustains fetal hemoglobin expression, significantly improving patient outcomes. Genome editing offers an innovative approach to treat V-thalassemia and sickle cell disease by targeting the erythroid-specific enhancer of the BCL11A gene. This process suppresses BCL11A expression, allowing continued production of fetal hemoglobin instead of the defective adult hemoglobin. By using CRISPR-Cas9 technology in hematopoietic stem and progenitor cells, this strategy prevents the disease-causing hemoglobin switch and has shown remarkable potential for treating these conditions. Clinical trials have demonstrated the potential of CRISPR-Cas9-based genome editing in V-thalassemia and sickle cell disease. By modifying hematopoietic stem cells, researchers achieved high editing efficiency, leading to increased fetal hemoglobin production. Patients treated with this method became transfusion independent, with the sickle cell patient showing no vaso-occlusive episodes, highlighting the safety and efficacy of this cutting-edge therapy. In the next few slides, we will be discussing modification of the somatic genome by transplantation. Transplantation serves as a form of somatic genome modification, where donor cells retain their genotype and introduce wild-type genes or replace damaged organs. For example, liver transplantation addresses familial hypercholesterolemia, while bone marrow transplantation provides effective treatment for lysosomal storage disorders like Hurler syndrome. Hematopoietic stem cells are a cornerstone of transplantation therapies, effective in both immune deficiencies and lysosomal storage diseases. These cells deliver lysosomal enzymes, correcting abnormalities like those in Hurler syndrome. Cord blood stem cells offer additional benefits, such as greater donor tolerance and reduced risks of graft versus host disease, making them an invaluable source for transplantation. Liver transplantation remains the only effective treatment for many metabolic liver diseases. Conditions such as cystic fibrosis and A1 antitrypsin deficiency account for a significant proportion of pediatric transplants. Additionally, it is used for metabolic disorders like urea cycle defects and MMA, even in the absence of liver disease. Outcomes are promising, with a 5-year survival rate of 75% to 85%, improved quality of life, and correction of metabolic abnormalities. Transplantation has transformed the prognosis of diseases like Hurler syndrome. Early intervention prevents cognitive decline and corrects physical abnormalities. Cord blood stem cells provide a promising alternative to bone marrow, with their reduced risks and wider availability. The future of transplantation lies in expanding the scope of stem cell therapies to address more genetic conditions effectively. Despite its successes, liver transplantation faces significant challenges, including complications from immunosuppression and a limited supply of donor organs. The future of transplantation lies in innovations like tolerance-inducing therapies and gene-edited stem cells. These approaches offer the potential to overcome organ shortages and improve outcomes by repopulating damaged tissues with genetically corrected cells. The next few slides we will discuss gene therapy options. Gene therapy is a groundbreaking technique aimed at treating genetic diseases by introducing functional genes into the patient's cells. Most often, this involves correcting loss-of-function variants, which are responsible for a majority of inherited disorders. Techniques like viral vector-mediated gene transfer or genome editing are at the forefront of this approach, offering hope for conditions like SCID and hemoglobinopathies. Gene transfer can be performed ex vivo or in vivo, each with unique advantages. In ex vivo therapy, cells are harvested, modified in a controlled environment, and then reintroduced into the patient. In vivo approaches, on the other hand, involve directly delivering the therapeutic gene into the body. Advances in vector engineering allow precise targeting to specific cell types, ensuring safety and efficacy. Choosing the right viral vector is crucial for the success of gene therapy. Retroviral and lentiviral vectors are preferred for their ability to integrate into the host genome, providing long-term gene expression. However, lentiviruses have the added advantage of working in non-dividing cells. AAVs are particularly valuable for their episomal stability and minimal immune response, while adenoviruses are ideal for larger genes despite their limitations in safety. While gene therapy holds immense promise, it's not without risks. Adverse immune responses, insertional mutagenesis, and potential germline modifications are key challenges. However, advances in vector design and regulatory frameworks aim to minimize these risks, ensuring that the benefits far outweigh the potential harms for patients with no other recourse. The future of gene therapy is incredibly bright, with ongoing advancements in vector design and CRISPR-mediated gene editing leading the way. From hemoglobinopathies to cancer, the scope of gene therapy applications is rapidly expanding. However, it's critical to ensure ethical oversight and equitable access as these therapies become more mainstream. Gene therapy has transformed treatment possibilities for over 24 single-gene diseases, ranging from retinal and hematopoietic disorders to liver protein deficiencies. These breakthroughs demonstrate how targeted therapies can restore function and improve quality of life for patients. For example, therapies for X-linked SCID and hemophilia B highlight the potential of gene transfer to correct underlying genetic defects effectively. X-linked SCID represents one of the most groundbreaking successes in gene therapy. By correcting the IL-2R gene mutation in stem cells, patients experience restored immune function and significant clinical improvements. However, early trials revealed risks such as insertional mutagenesis, underscoring the importance of refining vector designs to enhance safety. Spinal muscular atrophy marked a significant milestone in gene therapy for neurological disorders. By leveraging AAV9's ability to cross the blood-brain barrier, systemic delivery of the SMN protein provided life-changing outcomes for affected infants. However, challenges like hepatotoxicity and immune responses highlight the need for ongoing monitoring and innovation. Precision medicine exemplifies the future of treating Mendelian disorders, where therapies are uniquely tailored to the individual. By identifying specific genetic sequences, gene therapy can correct or mitigate disease effects at the molecular level. Advanced approaches such as allele-specific oligonucleotides, siRNA, and small molecule chaperones expand our toolkit for addressing genetic disorders. With these advancements, precision medicine holds the promise of transforming lives, offering longer and improved quality outcomes for patients worldwide. This is the end of this chapter. I apologize that I have had to use AI voice and avatar for the purpose of my PowerPoints moving forward. I have had technical issues with my computer recording my voice,